This is an example of an epinephrine signal transduction pathway. First, you have an RTK membrane receptor embedded in the cell's phospholipid bilayer. You also have an embedded enzyme known as adenylate cyclase. In the cell, you have a GDP attached to a G protein. Epinephrine will enter, and because it's hydrophilic, it can't cross the hydrophobic plasma membrane, so it will bind to the membrane receptor. This activates the G protein and it replaces its GDP with a GTP molecule. The G protein will then encounter adenylate cyclase, which will become activated. Then, ATP molecules will be catalyzed into CAMP molecules by the adenylate cyclase. Then, it will bind with the protein kinase A molecule, phosphorylating it, thereby activating a nearby phosphorylase molecule that will convert glycogen and a phosphate into a glucose molecule that will diffuse out of the cell. For the sake of the video, we just showed what happened close up with one CAMP molecule activating a protein kinase A. However, in reality, there are many. This is called amplification, and we demonstrate it here. Because this video demonstrates how epinephrine triggers glycogen breakdown in liver cells, once the glucose diffuses out of the cell, it enters the bloodstream so it can spread throughout the body. Like we talked about, in the liver cells, epinephrine will break down glycogen and release glucose into the bloodstream. In heart cells, it will increase heart rate, contraction forces, and the cardiac output. In smooth muscle cells, it will relax your muscles. Lastly, in adipose cells, also known as fat cells, it will break down triglycerides and release fatty acids as energy.